This module, describing the structure and functions of the nephron, is a part of the paper on human physiology. It describes the structure of the nephron and its functional organization. The learner will be able to describe the structure of the nephron, comprehend the different parts of the nephron, define, comprehend and relate the structure to the functions. This module aims to explain the structure and parts of a nephron, relate the structure to its functions. What is homeostasis? It is the self-regulatory mechanism in the body by which the body maintains the milieu interior or equilibrium inside the body. The urinary system is very important because it performs the essential function of getting rid of metabolic wastes. Now, let us study about the different organs of the urinary system. The main structures that make up the urinary system are the two kidneys, which are two bean-shaped structures. Each kidney contains 1 to 1.3 million nephrons, 2 ureters, 1 urinary bladder, 1 urethra, arteries and veins. The ureter connects the kidney to the bladder. The bladder is the storage organ for urine. When the bladder is sufficiently full, the person experiences the urge to urinate. Thereafter, urine is excreted to the outside of the body through the urethra. Here, you can see a diagram of the urinary system in the body. You can see the kidney on both sides, the ureter, the urinary bladder and the exterior opening, the urethra. Can you locate the two kidneys placed symmetrically on each side of the body? The ureters which come out from each kidney and the dome-shaped urinary bladder in the center. Can you also locate the adrenal glands on top of each kidney? Since they are found on the top of the kidney, they are also called as the suprarenal glands. In the next diagram, you can see the different structures that form the urinary system and how they are located in the abdomen. The urinary system consists of the renal arteries, the kidney, the renal tubule, the urinary bladder, the ureters and finally the urethra. On the right, you can see the longitudinal section of the kidney and the adrenal gland, the adipose capsule, renal medulla and cortex, renal pelvis, the two ureters, the sectioned urinary bladder which shows the detrusor muscle of the bladder. You can also see the openings of the ureters. Further down are the bladder outlet and the urethral opening. You can also see the blood vessels to the kidney. The renal artery branches out from the aorta and supplies oxygenated blood to the kidney, while the renal vein collects deoxygenated blood from the kidney and drains it into the inferior vena cava. Now, let us move on to the internal anatomy of the kidney. In the diagram, on the left is the photographic representation and on the right is the diagrammatic view of the kidney. You can see the three distinct regions of the kidney, namely the cortex, the medulla and the renal pelvis. You can also see the major and minor calyces which collect urine and empty it into the renal pelvis. The next aspect to be considered is the blood and nerve supply to the kidney. Blood supply to the kidney is through the renal artery and from the kidney through the renal vein. That is, blood supply into the kidney progresses to the cortex through renal arteries to segmental, lobar, interlobar, arcuate and cortical radiate arteries and back to renal veins from cortical radiate, arcuate and interlobar veins. The renal plexus regulates renal blood flow by adjusting the diameter of the renal arterioles and influencing the urine forming role of the nephrons. Now, let us move on to the functions of the kidney. The main function of the kidney is to filter fluid from the blood and concentrate the solution of waste materials which is passed out as urine. Besides urine formation, the kidney has the following functions. It has a role in regulating blood volume because it controls the amount of water to be excreted and the amount of water to be reabsorbed. It regulates electrolytes in the blood by controlling the secretion and reabsorption of sodium and potassium ions. It also regulates the pH of the blood by controlling the secretion and reabsorption of hydrogen ions. When more hydrogen ions are excreted from the blood, it renders the blood less acidic and more alkaline. But if more hydrogen ions are retained in the blood, this renders the blood more acidic and less alkaline. The kidney also regulates blood pressure by regulating the amount of water excreted and the amount of water reabsorbed back into the blood. When the kidneys excrete less water and reabsorb more water, the blood volume will increase. An increased blood volume will lead to an increased blood pressure. On the other hand, if the kidneys excrete more water, the blood volume will reduce. This will lead to reduced blood pressure. The kidney also plays a role in the regulation of erythropoiesis, that is, red blood cell production. When the number of red blood cells decreases, the level of oxygen in the blood will also decrease. This causes the kidneys to secrete a substance called erythropoietin. Erythropoietin travels to the bone marrow and causes it to produce more red blood cells. When enough red blood cells have been produced, this process is shut down via a negative feedback mechanism. Now that we have an idea about the renal system, let us zero down to the fundamental unit of the kidneys, namely the nephrons. The nephrons are the structural and functional units of the kidneys that carry out the various processes that form urine. They are responsible for filtering the blood and concentrating the solution to produce urine. In the process, macronutrients, electrolytes, and the required amount of water are retained, while waste substances, excess electrolytes, micronutrients, and toxins are expelled. As I have already mentioned, each kidney has 1 to 1.3 million nephrons. The number of nephrons begins to decrease after 45 to 50 years at the rate of 0.8 to 1% every year. Each nephron has two parts, renal or Malpighian corpuscle, composed of a tuft of capillaries called the glomerulus, surrounded by a glomerular capsule or the Bowman's capsule, and a tubular portion called the renal tubule. You can see the diagram of the renal corpuscle or Malpighian corpuscle here. The renal corpuscle is a spheroidal and slightly flattened structure with a diameter of about 200 microns. The renal corpuscle is formed by two portions, namely the glomerulus and the Bowman's capsule. The glomerulus is a tuft of capillaries enclosed by the Bowman's capsule. The capillaries are interposed between afferent arteriole and efferent arteriole. The afferent arteriole divides into 4 or 5 large capillaries, each of which subdivides into many small capillaries arranged in irregular loops. This is the first part of the nephron where fluid is filtered from the blood. It has two parts, the network or tuft of capillaries connected to an afferent arteriole at one end and an efferent arteriole at the other end, and transports blood to the site or the glomerular capillaries. The vascular system in the glomerulus is purely arterial. The second portion is an enlarged head of the nephron which collects the filtered fluid. This part of the nephron lies in the renal cortex. It is lined by thin epithelial cells which allow for fluid and other substances in the blood to easily pass into the nephron. The capsule is formed by two layers, the inner visceral and the outer parietal layer. Its function is the filtration of blood, which forms the first phase of urine formation. The functional anatomy of Bowman's capsule can be compared to a funnel with a filter paper. The diameter of the Bowman's capsule is 200 microns. The Malpighian or renal corpuscle is situated in the cortex of the kidney, either near the periphery or near the medulla. Based on the situation of the renal corpuscle, the nephrons are classified into two types, namely cortical nephrons or superficial nephrons. These nephrons have their corpuscles in the outer cortex of the kidney near the periphery. About 85% of the nephrons are cortical nephrons. Next type is the juxtamedullary nephrons. Nephrons with corpuscles in the inner cortex near medulla or corticomedullary junction are called juxtamedullary nephrons. We will deal with the juxtamedullary nephrons or juxtaglomerular apparatus in a short while from now. Let us continue with the next part of the nephron, namely the renal tubule. The tubular portion of the nephron or the renal tubule, as you can see from the diagram, is the continuation of the Bowman's capsule and penetrates into the medullary region. It is made of three parts, namely proximal convoluted tubule or PCT, loop of Henle LH. The loop of Henle consists of descending limb, hairpin bend and ascending limb. The last portion is the distal convoluted tubule or DCT. The proximal, distal and the collecting tubules are located within the renal cortex, while the loop of Henle is within the medulla. The first part of the collecting tubule is within the renal cortex, while the later part is in the renal medulla. Now, let us deal with each one of the parts one after another. The proximal convoluted tubule is the coiled portion of the renal tubule arising from the Bowman's capsule. It is situated in the cortex. It is continued as a descending loop of Henle. It is 14 millimeters long and has a diameter of 55 microns. Next is the loop of Henle. The loop of Henle consists of the descending limb, which has two segments. The first is a thick descending segment of the loop of Henle. It is a direct continuation of the proximal convoluted tubule. It descends down into the medulla and has a length of 6 millimeters and a diameter of 55 microns. The next part is the thin descending limb. The thick descending segment of Henle's loop is continued as the thin segment and it connects with the hairpin bend of the loop. The hairpin bend is continued as the thin descending segment, which opens again into the thick ascending segment. The length and the extent of the thin descending segment, hairpin bend and thin ascending segment vary in different nephrons. In cortical nephrons, it is short and the hairpin bend penetrates only up to the outer medulla. In juxtamedullary nephrons, this is long and the hairpin bend extends deep into the inner medulla. In some nephrons, it even runs into the papilla. Next, we move on to the hairpin bend. It is formed by flattened epithelial cells without brush border and is continued as the ascending limb of the loop of Henle. The ascending limb or the segment of loop of Henle has two parts, thin ascending segment and thick ascending segment. The thin ascending segment is the continuation of the hairpin bend. It is also lined by flattened epithelial cells without brush border. The total length of the thin descending segment, hairpin bend and thin ascending segment of the loop of Henle is 10 millimeters to 15 millimeters and the diameter is 15 microns. The thin ascending segment is continued as the thick ascending segment. The thick ascending segment is the continuation of the thin ascending segment. It is about 9 millimeters long with a diameter of 30 microns. It is lined by cuboidal epithelial cells without brush border. The terminal portion of the thick ascending segment, which runs between the afferent and efferent arterioles of the same nephrons, forms the macula densa. Macula densa is a part of the juxtaglomerular apparatus. The thick ascending segment ascends to the cortex and continues as the distal convoluted tubule. The distal convoluted tubule is the continuation of the thick ascending segment and occupies the cortex of the kidney. It opens into the collecting tubule. The length of the distal convoluted tubule is 14.5 to 15 millimeters and a diameter of 22 to 50 microns. The final portion is the collecting duct. The distal convoluted tubule continues as the initial or arched collecting duct which is found in the cortex. The lower part of the duct lies in the medulla. 7 to 10 initial collecting ducts unite to form the straight collecting duct which passes through medulla. The duct is made up of two types of epithelial cells, principal P cells and intercalated I cells. Now, let us understand the juxtaglomerular apparatus in some detail. We have already discussed that depending on the location of the nephron and the situation of the renal corpuscle, whether the renal corpuscle is situated in the cortex of the kidney, near the periphery or near the medulla, the nephrons are classified into two types: cortical nephrons or superficial nephrons and juxtamedullary nephrons. The cortical nephrons have their corpuscles in the outer cortex of the kidney near the periphery. In human kidney, 85% of nephrons are cortical nephrons. The nephrons which have their corpuscles in the outer cortex near medulla or corticomedullary junction are the juxtamedullary nephrons. Here you can see the juxtaglomerular apparatus, which is the specialized organ situated near the glomerulus of each nephron. The word juxta means near. It is a specialized structure formed by the distal convoluted tubule and the glomerular afferent arteriole. It is located near the vascular pole of the glomerulus and its main function is to regulate blood pressure and the filtration rate of the glomerulus. It consists of three parts: macula densa, extraglomerular mesangial cells and juxtaglomerular cells. The macula densa is the end region of the thick ascending segment before it opens into the distal convoluted tubule. It is situated between afferent and efferent arterioles of the same nephron. As you can see, it lies very close to the afferent arteriole and it is formed by tightly packed cuboidal epithelial cells. The extraglomerular mesangial cells or agranular cells, Lassus cells or Gurmati cells are situated in the triangular region bounded by afferent arteriole, efferent arteriole and macula densa. Now you may wonder what Gurmati cells are. They are modified smooth muscle cells primarily of the afferent arteriole of the renal glomerulus, discovered by Norbert, a Belgian physician, in 1890 to 1960. It is used synonymous with juxtaglomerular cells. Another type of mesangial cells are situated in between glomerular capillaries and are called as the glomerular mesangial or intraglomerular mesangial cells. These cells support the glomerular capillary loops by surrounding the capillaries in the form of a cellular network. They play an important role in regulating the glomerular filtration due to their contractile property. The juxtaglomerular cells or granular cells are cells in the kidney that synthesize, store and secrete the enzyme renin. They are specialized smooth muscle cells mainly in the walls of the afferent arterioles and some in the efferent arterioles that deliver blood into the glomerulus. In synthesizing renin, they play a critical role in the renin-angiotensin system and thus in the autoregulation of the kidney. Now, let us pass on to the details of renal circulation. The renal circulation is composed of highly specialized blood vessels. In adults, in resting condition, the kidneys receive 1300 ml of blood per minute, that is 26% of the cardiac output. Maximum blood supply to the kidneys enhances the functional efficiency of the blood. The renal blood vessels are composed of the renal arteries and the renal veins. The next figure shows the different regions of renal circulation. You can see the renal pyramid, interlobular artery, renal vein, the renal hilum, renal pelvis, ureter, minor calyx, renal capsule, inferior renal capsule, superior renal capsule, interlobular vein, cortical nephron, minor calyx, major calyx, renal papilla and the renal column. The renal artery arises directly from the abdominal aorta and enters the kidney through the hilus. It passes through the renal sinus and divides into many segmental arteries. The segmental artery in turn subdivides into interlobar arteries. Interlobar artery passes in between the medullary pyramids. At the base of the pyramid, it turns and runs parallel to the base of the pyramid, forming the arcuate artery. Each arcuate artery gives rise to interlobular arteries. The interlobular arteries run through the renal cortex, perpendicular to arcuate artery. Thereafter, the interlobular arteries branch into a number of afferent arterioles. The afferent arterioles enter the Bowman's capsule, divide into 4 or 5 large capillaries. Each large capillary further divides and forms the glomerular capillary tuft or loops. The capillary loops unite to form the efferent arteriole, which emerges out of the Bowman's capsule. The efferent arterioles form a second layer of capillary network called peritubular capillaries, which surround the tubular portions of the nephron. The renal circulation forms the portal system with 2 sets of capillaries, namely the glomerular capillaries and the peritubular capillaries. The peritubular capillaries and vasa recta. The tubular portions of the cortical nephrons are supplied by peritubulary capillaries and those of the juxtamedullary nephrons by the vasa recta, which are specialized capillaries. They are straight capillaries and hence the name vasa recta. The vasa recta arise directly from the efferent arteriole of the juxtamedullary nephrons and run parallel to the renal tubule into the medulla and ascend up to the cortex. The venous system. The venous system of the kidney is constituted by the peritubulary capillaries and vasa recta, which drain into the venous system. The venous system comprises of the peritubular venules, constitutes the interlobular arteries, arcuate veins, segmental veins and the renal vein. The renal vein leaves the kidney through the hilus and joins the inferior vena cava. This figure shows the venous supply to the kidneys. Next we move on to the aspect of regulation of renal blood flow. Renal blood flow is regulated by the process of autoregulation. There is no significant role for the nerves innervating the renal blood vessels. The process of autoregulation is the intrinsic ability of the organ to regulate its own blood flow. The phenomenon operates in some vital organs such as the brain, heart, and the kidneys. Autoregulation in the kidneys is important for the maintenance of the glomerular filtration rate. Thus, renal blood flow is normal even when the mean arterial blood pressure varies between 60 and 180 millimeters of mercury. This helps to maintain normal GFR. As you can see from the figure, the glomerular filtration rate is directly related to the volume of renal blood flow. 2 mechanisms are involved in renal autoregulation, namely myogenic response and tubuloglomerular feedback. Whenever blood flows into the kidney and the volume of blood flow increases, it stretches the elastic wall of the afferent arteriole. This increases the extracellular fluid into the cells. The influx of calcium ions leads to the contraction of smooth muscles in afferent arteriole, which causes constriction of the afferent arteriole. Hence, blood flow is automatically decreased. In tubular glomerular feedback, the kidney changes its own blood flow in response to changes in sodium concentration. The sodium chloride levels in the urinary filtrate are sensed by the macula densa cells at the end of the ascending limb. While sodium levels increase, the macula densa releases ATP and reduces prostaglandin release to the juxtaglomerular cells. The juxtaglomerular cells in the afferent arteriole thereafter constrict and the cells in both the afferent and efferent arterioles decrease their renin secretion. This lowers the GFR. If there is further increase in sodium concentration, nitric oxide, a vasodilator, is released. This prevents excessive vasoconstriction. On the other hand, if the juxtaglomerular cells are stimulated to release more renin, the renin-angiotensin system is stimulated to produce angiotensin II, which constricts the efferent arteriole and increases GFR. Thus, the kidney maintains its own blood flow. Finally, let us understand about the uniqueness of renal circulation. The renal circulation is unique in that renal arteries arise directly from the aorta, and the high pressure in aorta facilitates the high blood flow to the kidneys. Kidneys receive the second largest volume of cardiac output, that is 1.3 liters, 26%, next only to the liver, which receives 1.5 liters or 30%. Thirdly, blood is completely filtered at the renal glomeruli, which have a double network of capillaries, that is the glomerular and peritubular capillaries. The glomerular capillaries form a high pressure bed with a pressure of 60 to 70 millimeters of mercury. This augments filtration. The peritubular capillaries form the low pressure bed with a pressure of 8 to 10 millimeters of mercury, which helps in tubular reabsorption. Autoregulation is yet another special feature of renal circulation. To conclude, the nephron is thus a composite organ which purifies blood by filtering off the impurities and excretory substances from it. As the blood plasma flows through the nephrons, fluids comprising excess water, salt, and metabolic waste products are extracted from the blood as it enters the Bowman's capsule in the nephrons. The fluid in the Bowman's capsule is called glomerular filtrate. It is similar to blood plasma except that it has almost no protein. The fluid moves through the renal tubule. It is called as the tubular fluid. It's different from the glomerular filtrate because of substances which are removed and added by the tubule cells. Finally, when the fluid moves into the collecting tubule, it is called urine.